We have a saying at Amgen that scientists with AI are going to be better than scientists alone or AI by itself. And I think critical for that is for scientists, especially those who come from different fields in the therapeutic space, to really understand what makes AI useful and what makes it click. So I've been engaged in this process for the last year while I've been at Amgen. I've been very lucky to have incredible thought partners in this, including Kai-Yi Zhang, who is a Rice BSBE recent alum. He's an undergrad from the class of 21. These kids these days, it's crazy. He's also our inaugural Amgen Science Fellow. He'll be starting his laboratory at Princeton in bioengineering in next spring, but for the year we have him at Amgen. And he's been an incredible thought partner as we think about how we can use AI to accelerate drug discovery. We also have the team in Iceland at Decode Genetics, for which we have a localized superpod. This is where we do a lot of our heavy lifting in this space. And I've been working with Erico Silva there, a very talented mathematician from Princeton, to develop a lot of the core concepts that I'll share with you today. And lastly, a little bit about myself. I'm really appreciative to have this invite to come back to Houston. It was about 10 years ago, almost to this day, that I started my lab at Baylor College of Medicine. And I couldn't think of a more nurturing and collaborative environment for a young PI than here in the TMC. Many of my friends, colleagues, collaborators were here at Rice University. And I'll showcase a lot of work today, in particular from Caleb Bashore's lab, who really became a great thought partner and friend for me. We had many coffees together at Java Lava up the street before it closed during the pandemic. All right, so this is really the core thesis of what I want to talk to you today, especially for those of us who are not at those technical talks. The fuzziness in which we talk about AI really limits its practical utility. I think the onus is on us as drug developers and people trying to impact human health with AI to have a clear and precise understanding of what we mean when we talk about AI, because that's going to help us use it better. And so, similar to kind of the double entendre of my talk, I want to be precise in these definitions. When we talk about enabling AI in drug discovery, I really mean two different things. The first, what is enabling about AI right now in drug discovery? What AI applications are enabling and why? And the second is, what do we need to do next? What can we do in drug discovery to better enable more useful AI? And I'm really gratified to come after Payal's talk, because a lot of the things that she mentioned, I think we have found also at Amgen are really necessary to make the next leap. And so within this first part, I want to spend just a few minutes defining really what I think are the key criteria that underlie successful AI applications. And by understanding them, we can understand better in a more intuitive way where AI applications are going to be useful and where they're going to run into inherent limitations given, for instance, the state of the data or the way that the problem itself is formulated. Okay, so first and foremost, what we call AI are a set of machine learning applications that turn non-convex problems into locally convex problems. Science in a lot of the real world is like this. It's messy. There are a lot of local minima. From a mathematics standpoint, it's very craggly. It doesn't really fit well into transformations. And what we've realized over the last 20 plus 30 years in the machine learning field is that by transforming these craggly natural landscapes into locally convex representations, it gives us the ability to find optimization, pattern recognition, much more computationally efficiently. This really is the crux of why AI has been so successful, because we've been able to take complex biology, complex natural phenomena, and then transform it in such a way, and it has all of these very, very nice sort of corollaries to it. In addition to the optimization, the compute speed, it can also reduce, for instance, the number of spurious solutions. So this is really key. And this is, of course, sort of a high-level conceptual diagram. Let me give you a real-world example. When we think about protein-ligand interactions, historically, like in the molecular dynamics space, we think of it as an energy landscape, trying to minimize Gibbs free energy, thermodynamics. And that's really hard. You've had organizations like Schrodinger, like D.E. Shaw over the years trying to invoke quantum computing and a lot of these very computationally intense methodologies to do this using physical simulations. And I think this is very exciting and rewarding work because it gets at a more proper modeling of what's happening at an atomic level, but it's fundamentally computationally hard. And so one of the big breakthroughs that's happened early in the machine learning field is we can take these protein-ligand interactions and reformulate their representations, in this case, as graphs with nodes and edges. And graphs are really, really good for machine learning. If you can put it into a graph, it's kind of like step one. You're on your way to having a solution for which machine learning has the ability to optimize. And this is exactly what many approaches do today in multiple areas of drug discovery, but in particular in the space of protein-ligand interactions. And it allows you to, one, take this sort of complex atomic resolution, formulate into a graph, but then in stuff that's sort of gory, and I won't go into too much, encode a series of embeddings and, for instance, allow these nodes to pass information to one another. And this forms the basis of these graphical neural networks and message passaging neural networks that then allow us to optimize, learn, and ultimately predict protein-ligand interactions. And this has really been the core of a huge number of successes in the field. Okay, so that's one. The second is if you do that, it enables successful pattern recognition. My favorite machine learning example is from the hit TV show Silicon Valley, hot dog versus not hot dog. And in biology, that's a little bit harder. But when you can define attention really nicely, it can tell, essentially, the machine learning where to key in on. And so, for instance, this is an algorithm called Evolve Pro, which was developed by Kai while he was a graduate student at MIT. And I want you to focus on is, as it's trying to evolve this T7 RNA polymerase, the particular amino acid residues that it suggests the most number of changes to. Now, why is this important? Well, as I'll go into later, proteins are really crazy. They have an infinite theoretical design space, right? 20 amino acids, all the theoretical combinations. But yet in the natural world and in practical biology, they can tolerate very few changes. And it turns out more often than not that a lot of where you want to modify the activity of the protein converges into a small set of areas co-localized in three-dimensional space where essentially the action is happening. And in this case, that's exactly what they found, that they could recognize from sequence-based and activity-based models the sets of amino acids that basically corresponded to, in this case, structurally known and molecularly defined active regions of the protein, for instance, like this nexus between the protein and the nascent RNA. And so I think this is a really gratifying and key way to understand when AI is working, when the pattern recognition focuses you in on key points in the structure that are mechanistically active for the biology. And what I think has been beautiful to see with programs like ESM2 is that you can learn a lot of this just from the sequence space. That's really what's been transformative. You don't need to generate the crystal structures for everything. All right. So really, this gets us into what sort of we think of as three primary characteristics that make AI effective, especially in this space. Number one, a constrained feature space. Number two, the right training data. And number three, the ability to drive a reinforcement learning cycle for model refinement. So I'll go into each of these in a bit of detail with a few examples. All right, so as I alluded to earlier, proteins are pretty wacky. These are three seminal papers from the early 2000s as we sequenced the human genome and started to learn about the diversity of proteins across the evolutionary space that were really impactful in my thinking about it, and I think have read through to how our AI models work today. So the first is this paper from 2010, which postulated that proteins are evolving slower than they should. So if you think about a molecular clock, and they use this really funny example of the expansion of the local universe, if you think about proteins diverging over time through evolution, there's essentially a theoretical space that proteins can expand to, but in reality, there's a constrained space. And in many cases, for organisms that are divergent from many, many, many millions of years, you start to see now evolution, I wouldn't say hitting a dead end, but reaching constraints. So it's not just sort of infinitely expanding. Part of that reason is that most natural proteins can tolerate very few changes. So if you take, for instance, this beta-lactamase protein from this 2006 paper, there are very few amino acid substitutions that you can actually put into it that result in a protein of comparable fitness. And when those substitutions happen, it tends to put you down this cascade of only a certain number of additional allowable changes are permitted. And so when we think about protein design, especially using AI models, evolution is telling us that there are only a small number of amino acids that we can really mess with, and that most changes are going to completely disrupt the protein function. And this is also observable in reverse. So Jack Szostak's lab in 2001 did this heroic experiment where they synthesized essentially a gajillion random proteins and looked for activity, in this case, the ability to bind ATP. And they put this through what was an early version of directed evolution. But the reality is, if you just synthesize random proteins, essentially none of them have activity. And so this means if you think about a large theoretical protein space, there are essentially these very, very tiny local minima for which there's very little wiggle room around them. And this is where protein activity really lies. And this is great from the perspective of machine learning because it essentially implies constraint. And so this gets us into the training data. Now, this is a very simplified representation, but I think it kind of talks about the challenges that we have in biology. So if you wanted to design the world's most perfect machine learning kind of exercise, you would ideally have a scenario like this, where your training data was densely sampling some observed natural space, and that observed natural space was defined and existed within a larger theoretical space. Human language is perfect for this. We know the observed natural space. We could just look on Twitter, and we can sample it, and by and large, pretty much everything that has been said will be said by some bot online, so we've got that covered. Biology is a little bit more challenging. In some cases, we actually don't quite know what the theoretical space or natural space will be. We're flying a little bit blind. And then because of real-world challenges, technical difficulties in generating data, we often have training data that is highly biased, clustered, and localized. One key example of this, for instance, is that almost all of the kinase inhibitors that we use today, either in the clinic or in the lab, they share a very, very small number of core structures. And that's because they all came from early ATP competitive biochemical screens and were largely derived one after another. This is how drug discovery has worked for us. But this is not how you would design a machine learning training. And so we really have to then think about accounting for this fundamental weakness and how it's going to bias our molecules, or our models, excuse me. One key example of this, for instance, is that because the early AlphaFold versions were exclusively trained on structural data, they didn't have the ability to distinguish essentially a not protein versus a true protein found in biology, but one that was intrinsically disordered. And this was a limitation that was overcome by later evolutionary models that because they could be trained on protein sequences, which included proteins that you could crystallize versus proteins that just simply existed in nature, they were actually able to figure this out. Okay. And so this is, I think, a theme that will emerge over and over again. That when you have the right data, for instance, this is an example with AlphaFold of moving from structural to ESM with sequence and evolutionary-based data, you can actually do more and learn more. And not only is having the right data important, but the right quality of data is really important too. And what we've learned over the years is that high-quality data democratizes AI. And so here at Amgen, also working with the incredible group at Quebec and Mila, we developed a protein language model, Amplify. And for us, the really key hypothesis we tested is scaling necessary for performance? And the answer is no. By using a much more highly curated data set, we were able to train a model amplify that matched the performance of the 15 billion parameter ESM2 model with only 350 million parameters. And this was able to be trained pretty darn efficiently on a very small number of GPU nodes. And here you can see using this pseudo perplexity where to the left means better performance, better confidence of the model in predicting amino acids, that although ESM2 required multiple stepwise scalings to reach high performance, we were able to do it pretty quickly. The second thing, and this gets back to this idea that protein space is fairly constrained, this also creates a challenge in many of these machine learning algorithms where you always wonder, are you memorizing and just repeating back, or are you actually predicting? And one way to figure that out are looking at amino acid positions that are highly conserved, these lighter shaded bars, so basically you can't tolerate any change, versus amino acid positions that have wiggle room in the assorted genomes that were used to train these models. And for instance, in the case of amino acid positions with wiggle room, if you're overfitting, then you're always going to spit back the most common amino acid that's found in evolution, even if you're trying to optimize for some different activity criteria. In other words, you're not going to appropriately explore the space. And one thing we realized is that these models started going to very high parameter space. We started to see a decrease in the performance there. The number of times that they would basically just give you something different started to go down. Okay. So in addition to having the right data as a sort of training requirement, it's also really important to think about what you want to achieve in terms of the functional readouts and doing the experiments. And so this is, again, from Kai-Yi Zhang's paper, EvolvePro. And this, to me, has been kind of a really watershed moment as an experimentalist, because it says there are easy ways to greatly improve the performance of these models. You just have to pick up a pipette. So we talk about zero shot a lot in machine learning. And that's really the ability of a model to predict something and to essentially perform in a case where it's seen no relevant training data. And by and large, I think the fact that we can actually have any zero-shot activity is like a wonder, but it's still not that great. And so kind of the next logical thing to do is, well, let's just take our models and refine them and fine-tune them based off of a small amount of data. And you can do this in sort of a pre-training space. And here, Kai shows that as you escalate the number of mutants that you put in a pre-training that have sort of different levels of activity, then the models now perform much better. But there's even something simpler you can do, which is to go through this reinforcement learning and refinement cycle. Kai showed that if you just go through 10 rounds of model refinement, where in each case you actually make a small number of candidates predicted by the model and test them, it allows you to achieve performance far greater than the zero-shot models and equivalent to a lot of pre-training. And so this is great. It suggests that as we try to improve these AI models, we don't need tons and tons of data. We don't need the large data scales that we've normally used in model training. Even 10 to 15 additional experiments can make a huge difference. And then lastly, it's really important to know what you're optimizing for. If you take a model like ESM2, which is essentially optimizing on evolutionary fitness, you're going to get one kind of optimization landscape that your algorithms will traverse through. And maybe that's what you want, but maybe it isn't. In a lot of cases in drug discovery, we want to optimize for a particular activity. And really, I think this is a key realization. There's no good way to get there unless you encode that in such a way that the machine can compute on it. And so in this case, you have to have activity data in order to give yourself the ability to optimize on an activity landscape. So for those of us worried that AI is going to take our jobs in the wet lab, I think this is pretty reassuring. We still have work to do. And the right work can really make these models much more efficient. So this is how you can get there. One, I think, under-leveraged utility of machine learning these days is to help inform us in the wet lab what are the best next set of experiments to do that will give us the biggest increase in model performance. And so Caillou was able to basically train a model to do this, and it would take from what it was observing in the sort of gradient optimization and return to you a small number of variants that said, hey, if you measure this, we'll have a better ability to then train and refine our models. And this, I think, is the way that we should think about this sort of nexus between the wet and dry lab. A lot of our dry lab experiments need to turn into real-world experiments, and models like this can really help us. All right, so I'll get into the last part of my talk. And here, we really want to start thinking about what do we need to do next. So better training data gets covered a lot. We won't cover that today, but I think it's an obvious case where the better data we have, the better our models will be. Instead, I'll talk about two things that don't get covered a lot, but are very important. Better attention. I have a 2-year-old, so this is very important, as well as having a better reinforcement learning cycle, a little bit of what I alluded to with Caillou's work. So first and foremost, humans aren't innately good at drug discovery. The fact that we've succeeded at all, I think, is a triumph and a testament to our perseverance. But we can't look at these two molecules right here in this room, well, most of us can't, and know which one's the drug and which one's not the drug. Does anyone recognize these two molecules anywhere? Okay, good. Point proven. This is imatinib, of course, one of the most famous drugs in all of human history. And this is tubocurare, the poison dart frog toxin. But this is also a trick question because apparently in the 60s we used this as a drug too. Don't ask me why. And so, whereas in a lot of the machine learning and AI applications that we experience in the kind of consumer world, like hot dog versus not hot dog or doing CAPTCHAs for Google, humans don't have an innate ability to look at complex things like chemical structures and be able to understand functional properties like which one is a drug or which one might bind a protein. And so this really starts to challenge how we think about and define things like attention. Now, most machine learning models and algorithms do have some attention-based mechanism in them. And so my point is not to introduce them. My point is how do we know that they're right, and how do we know that the attention is pointed in the right direction? In the case of image recognition, it's very easy to validate and see these things. Here are a set of pictures of cars, and the right, this is what the machine learning is telling us it's paying attention to as it takes these different keywords and basically figures it out. And of course, you see things like if you have the make and model of a car, it's going to focus on the decal. And for some reason with Lambos, this little front hood is the iconic shape. Go figure. I've never had a Lamborghini, so I wouldn't know. But this is what biological data often looks like to us. I'm sure many of you from the genomics systems biology field are familiar with this. It's the classic hairball effect. And Eriko in Iceland had this nice quip. It's all there and yet somehow useless. I'm sure we've all stared at gratuitous heat maps and hairballs. And it's tricky, right? So the data itself is highly biased. We have lots of false positives. Proteins stick together and tend to get recorded as PPIs in large databases. There's a massive publication and study bias. Everything interacts with MYC or P53 for some reason. And we tend to have a technological bias. We sequence everything, and so we're overrepresented for sequencing-based readouts. And so although we can encode biological information in these machine learning amenable graphical formats like knowledge graphs, it's very difficult for us to know what's important and what's not. Historically, we've accomplished this using different approaches from network theory and stuff like that. And one very common one is to look for network motifs that might be biologically relevant and understand whether or not they're overrepresented in randomized networks or non-relevant networks. But for this to work, it requires you to binarize the data, so you make these cute little arrows. It only works when there's a small number of data layers. If your edges have many layers to them, it's going to get too complicated. You need lots of true biological networks to serve as your references. And then it's fairly computationally horrific because of the combinatorial complexity as you go from 3 to 4 to 5 to 6 and subgraphs. So what do we need in this space? Well, we, again, and Payal really nicely alluded to this, we need to move from ways of passing messages and information just from the nodes to also the edges, and we need to use this as a better way to define attention in these biological knowledge graphs. And this has worked reasonably well for protein ligand structure, but I think for biological networks and functional networks, this is still an emerging idea. So you can imagine a node X that's influenced by a set of upstream nodes in this ensemble psi. And each of these nodes is going to pass information to X via sort of a multidimensional embedding. And then critical to understanding biology, we're probably missing stuff, or many of these nodes are probably going to be false positives. And when these turn from simplified toy diagrams to those big hairballs, it's very difficult for us to know what's important and what's not. But if you can design your biological networks in this way, it sets up a very simple sort of explanation, where X hat, for instance, is the predicted contribution of these nodes to what you observe in X. And you can look at the difference between those things, and that starts to tell you, hey, does this set of nodes explain X? If so, that's great. It will tell you within it which edges are giving you the highest contribution. And if that isn't the case, then not only can you say, hey, this doesn't explain X, but you can start to postulate what kind of missing information you might have. And we need these kinds of things in biology, given the messiness of our data, given that we have lots of biases in how it's generated. We cannot assume, for instance, that we're uniformly sampling the relevant biological space. And this gets into something that I think has been very popular these days, but what is old is new again and vice versa, this idea of the virtual cell that many are talking about that I think will become a triumph of this field. But all the way back in 1970, these two mathematicians, Roger Connon and Ross Ashby, they postulated this very, I think, prescient idea. Every good regulator of a system must be a model of a system. Okay? So what does that mean? It's actually really, really cool if you think about it. All right? The design of a complex regulator often includes the making of a model of the system to be regulated. This is really cool. In this paper, a theorem is presented which shows under very broad conditions that any regulator that is maximally both successful and simple must be isomorphic with the system being regulated. So essentially, everything has to be a model of itself in order to be a good regulator. And by and large, we're seeing that the cell itself is a good regulator. The way in which the cell actually operates, the way it senses, responds to, and transduces information is itself a model of, for instance, environmental sensing. I'm sure many of you have seen these kinds of network diagrams that have by and large turned out to be true for cells, and it essentially is a model of the system. We take external cues, integrate them, and that leads to readouts and activities. And so if the cell acts this way, where it's a good regulator, then what we really need to be thinking about is, does the virtual cell act as a good regulator? And to date, we're not doing this, in large part because the virtual cell is still, in many cases, a black box. And so we can take a virtual cell and we can predict things and we can validate the predictions. And this tells us whether or not we're pattern matching. But it doesn't tell us whether we've attained a sort of approximation of the good regulator theorem. And I think this is a really important avenue that we have to go down in these virtual cell efforts. How do we ensure that the cell is a good regulator? Okay, so in the last few slides, I'll then get back to the wet lab. How do we refine our models and have a good reinforcement learning cycle? So this is how we think of a lot of reinforcement learning in modern ML and AI applications. There's some kind of agent, there's a feedback loop, there's either an external or internal critic, shall you say, and this gives a reward mechanism, a little cookie to the agent if it's producing what you want. And this underpins most successful applications in AI, and biology is no different. The difference in biology is that this cycle spans the wet to the dry. How we generate the data, how we validate the data, and how we refine the data, really those need to be novel data generated through experimentation. And critical to this, the kind of data you want for training is not the same kind of data you would want for validation, and is not the same kind of data you would want for refinement. And where we've done pretty good at this and this, it's here that I think we need better experimental systems. So luckily, they're making them upstairs, and we are really excited about this work. And this is a very, very elegant system from Caleb Bashor's lab, what they call Classic, which essentially allows you to do combinatorial assembly of different DNA elements into these circuits and to assess their activity in a highly controlled and pooled manner. These are the exact kinds of experimental systems that we need as we move towards an AI-driven future. When you have these kinds of systems, it allows you to kind of not only reimagine the classic design, build, test, learn cycle in synthetic biology. I would argue that, for instance, this perspective that they wrote should be flipped the other way around. It gives us the ability to enhance and accelerate our machine learning. We talk about space a lot in this talk, and you think of traditional synthetic biology as being sort of this random wiggle moving towards your optimization. But if you can properly learn the design space and then design a set of features that you want to test to explore it, you're going to have a much better opportunity to really get to your optimized criteria. And so that's exactly what they've shown that this experimental platform can do. They've used it to generate these beautiful cell circuits looking at multiple transcriptional components and the effects of those on output, both in a basal and an induced setting. And this is really cool. You can see here that they can explore a very large space in terms of their measurements, and that they can use this to train ML models that can basically recapitulate that space to a high degree of accuracy. And so when you then think about this and where you take the next step, it's all about refinement. Where is an interesting parameter space? In this case of high fold change circuits, these are the kinds of places where you want to understand your predictions and how that varies from the data, and then build and test the next generation of reporters, or in this case, circuits, to read that out. And so to do this, we really need these kinds of programmable, methodical approaches to do this in a very rigorous and systematic way, controlling for all other variables except for the ones you want to change. And we need to do this efficiently. Another way we can do this efficiently, and again, this is from Kai-Yu's grad work, he had a much more productive grad school than I did, is using RNA sensors. RNA, thanks to the tremendous progress of the pandemic and the vaccines we created, is now a much more facile tool for therapeutics, but also for research technology. And we can now start to create these RNA-based sensors that we can order, deliver via lipid nanoparticles, and all of a sudden you get to live cell imaging with high accuracy gene expression in a way that doesn't require any cell line engineering or ghoulish tinkering. For those of us who remember lentivirus and making things like MS2 hairpins and doing fish, this is remarkable stuff. So I'll just close by summarizing what I've talked to you today about. First and foremost, successful AI relies on these three features: constrained feature space, good training data, and the ability to refine and reinforce. It's pattern recognition at its heart, this ability to regularize non-convex landscapes into something that, for instance, a graphical neural network can optimize. And the key to it is making sure the activity that you want is actually encoded in these models. It will always find what you tell it to find, but you've got to give it a way to get there. And then secondly, as we start to improve these AI models, we've got to get better ways of prioritizing and defining attention that's relevant for biology. And we've got to start incorporating into kind of our feedback loops experimental systems that are really amenable and fine-tuned for this specific task. And I think if we do that, we're going to make a lot of progress in the coming years. So this is an area that I'm really optimistic about, and I hope I've conveyed some of that optimism to you today. Thank you.
Any questions? Hold on, it's a long walk with the mic, but I think we're streaming this, so we need the mics.
Hi, very nice talk. Thank you for that. So I have a question regarding your virtual cell, like the slide regarding that. So I was interested in the virtual cell models, for example, state, etc., that are currently available. And you also talked about like the data, quality of data, or let's say good training data. So to actually develop something like a programmable cell or virtual cell, how accurate the data has to be, and also corresponding to wet lab studies, like the data that we train on to, work on the virtual cell competition, let's say. How do you know that that's good data?
It's a very good question. So just to reframe the question, in kind of building virtual cells, how accurate do I think the data needs to be, and how good is it? So I think the trend has always been the higher quality your data, the better your models perform. I think one thing that we need to think about is that much of our data in this space is highly biased towards sequencing-based readouts. I'll take gene expression as an example. And often at longer time points, like 2-3 days. And if you go back to the very early gene expression papers from like Aviv Regev and others, there is this finding that multiple distinct perturbations at longer timescales all converged on a conserved gene expression program. And this is true biology, and that program was called the integrated stress response. And so you want to make sure that you're not simply pattern matching to that. So I think the question may be better reframed as not only how much and how good of data you need, but what kinds of measurements are we likely missing? Dynamic measurements, I think, are essential. If you take that same analogy of long-term gene expression data, and it all kind of muddling together after 24, 48 hours, if you look at nascent transcription and those changes at 6 hours, you can then very clearly differentiate different biological perturbations. So I think we need those different kinds of data and ultimately different mechanisms. There's so much of what's happening in the cell that we fundamentally can't measure. Glycosylation, we're really bad at that right now. But that's obviously important, and that's, I think, something that will be a continuing limitation of these virtual cells until we generate the requisite data in that space. That's a great question.
You mentioned maybe building another model or a process by which to identify the missing information, or if we get that one more piece of information, we could get this much more performance. One, can you expand on that, how you did that a little more? And then two, can you think of any other applications of that process kind of in other spaces?
Yeah, I think this is still very early stuff for us. It's really just about, and I think Payal really hit the same idea, is that historically we're not doing this sort of mutual information passing through the edges themselves. But the edges are where all of the activity happens. And so when we think about some of these mechanisms, I think just training on and understanding what are the missing information pieces, that's a very vague idea right now. But what we intend to do with it is to really think about different layers of information. So protein-protein interaction network, post-translational modification, RNA localization, stuff like that. And if we think about building these regulatory networks, if one of those mechanisms or the totality of those mechanisms can't explain the observed phenomena, then that tells us we're probably missing like an entire new edge type. That's, I think, the key for us. So it's really saying to us, hey, where is there missing biology beyond the set of mechanisms that you've looked at? And that's going to eventually provoke us to do the right experiment, hopefully. The flip side of that is it should then, when it's working, tell you that, okay, the main reason we're able to explain the observed data is primarily through protein phosphorylation or something like that. And as a drug developer, I think that's going to be very helpful because that's going to, again, focus our refinement and our own experimental validation and say, okay, now let's go generate, for instance, the phosphoproteomics data. So that's really what we're using it for, is to think about edge types primarily, where either the data is explanatory or it's missing.